So welcome back. We will start the second session with a talk by Professor Jonathan Marchant and Dr. Gihan Gunaratne. Jonathan is the chair of the Department of Cell Biology at the Medical College of Wisconsin. His research group has a long-standing interest in the pharmacology of calcium signaling pathways, with a focus on the druggability of calcium channels for treating neglected diseases. And today he is here with Gihan, a postdoctoral researcher in the Marchant lab. Gihan completed his PhD at the University of Minnesota in 2018, working on NADP-evoked calcium release and its role in regulating coronavirus infectivity, as well as the cell biology of TBC interactors. Now, during his postdoc, he has been working on identifying NADP binding proteins using a photoaffinity labeling approach, which he will talk to us about today. In fact, this first team talk will be on the use of a novel biofunctional photoprobe to identify NADP binding proteins. You can begin.
Great. Thanks, Martina. Well, it's really exciting and also somewhat amazing to have so many participants at this first ever NADP binding protein meeting. So good afternoon, good morning, everyone. These are team talks, so my job is simply to introduce our team. First and foremost, I want to give huge credit to Tim at the University of Minnesota, who's really been the driving force for all this work. He's really been at the epicenter, as we know, of this field, bluntly, since the field began. So we're all very grateful for that help you've given us. Jim Slammer at Toledo, who's our med chemistry innovator, without which we would have no tools at all to play with. And then Sandip and Eugene, who've been invaluable long-term collaborators with functional assays and ideas. Really, most credit must go to those who have been on the experimental front line. So I'd like to introduce Yaping and Gihan, who are two PhD students in my lab who worked successively and also successfully on this project. Gihan, who's now a postdoc, is going to give our talk today. So for us, as has been introduced already, this all really started when Yaping and Tim published results with the first generation photoprobe in 2 JBC papers way back in 2011, so a whole decade ago now. And the provocative conclusion at that time, which is now consensus, is that NADP doesn't directly activate TPCs, but binds to these elusive accessory proteins. And their work showed that this principle was also true in sea urchin. That was work with Tony and Gita, who talked first today. And I'd also like to acknowledge Andreas's work in T lymphocytes, or another primary cell type. All this body of data pointing to the existence of these low molecular weight NADP binding proteins. So overall, this has really been a team science effort to design, iterate, and optimize new probes. And Gihan is going to show you some data about 2 recent breakthroughs, which required first a new photoprobe, and then secondly, as often in science, some good luck finding the right cell type for a purification effort. All this is Gihan's work, so I'm simply going to hand over to him.
Great. Well, thank you for the introduction, Jonathan. My name is Gihan Gunaratne, and so as Jonathan had mentioned, this has been a sort of ongoing effort for about a decade now. And so today we're really excited to show some of the progress that we've made, and that's been through the use of a novel bifunctional photoprobe to identify NADP binding proteins. So starting with an outline, so I'll show you in the first part of my talk our probe-based purification of NADP binding proteins. And this was really enabled by 2 key developments, the first of which was the invention of a new bifunctional photoprobe that we call all-in-one-click, or AOIC, NADP. The second development was the somewhat happenstance discovery that erythrocytes are actually a very convenient biological source for NADP binding proteins. So these 2 developments combined allowed us to identify a new putative NADP binding protein, which in the second part of my talk, we will functionally validate and characterize, and that will come in 3 ways. First, at the level of the molecule, we'll look at ligand binding. Then at the cell signaling level, we'll look at calcium evoked by NADP. And then at the cell biology level, we'll look at endolysosomal trafficking, which is, of course, a process that's canonically associated with NADP signaling. Okay, so first introducing our photoprobe, AOIC NAADP. So this is a bifunctional probe that was invented and synthesized by Professor Jim Slama and his student Si-Jun He at University of Toledo. So here's the chemical structure of our photoprobe, AOIC NAADP, and there's really two functional moieties that I want you to keep in mind. So the first is this aryl azide group shown here. And upon UV irradiation, this aryl azide will cross-link to adjacent proteins, or in other words, we can form a covalent bond with unidentified binding proteins. And the second group is this alkyne moiety shown here. And these alkyne groups are amenable to copper-catalyzed alkyne azide cycloaddition reaction, or otherwise known as copper click reactions. So here we have a biotin azide group, which we're able to use to biotinylate a probe protein complex. So using this probe, we're able to essentially biotinylate unidentified binding proteins and then use biotin streptavidin interactions to purify these unidentified proteins. Now, as sort of prerequisite for the usefulness of any new probe is that this probe must still retain its ability to bind its targeted receptor. So to test whether our AOIC photoprobe is still able to interact with NADP binding proteins, we tested it functionally in microinjection experiments. So here are single isolated SKBR3 cells. This first panel shows injection with unmodified NAADP. So we see a rapid, robust NAADP response shown here. And similarly, when we inject our bifunctional probe, we get a response of similar magnitude. Now, importantly, if we were to pre-incubate these cells with baflamycin, the vascular ATPase inhibitor, these responses are significantly blunted. So that really suggests that this probe is still able to mobilize acidic calcium stores. Now, the next big breakthrough that we had mentioned was the realization that human erythrocytes are showing very intense and clean labeling of that 23 kilodalton binding protein that we've been chasing after for about a decade. So this is a photo labeling experiment, and here we've incubated lysates with P32 AOIC NAADP in the absence or presence of unlabeled NAADP. So this is comparing lysates from our erythrocytes versus two different NAADP sensitive cell lines, namely Jurkat cells and U2OS cells. And you can see here that we have nearly a threefold heavier labeling of this 23 kilodalton band. And this labeling was selective. We tried to protect this binding site using high concentrations of various nucleotide analogs, and as well as high concentrations of different calcium mobilizing second messenger molecules, cyclic ADP ribose and inositol triphosphate. And nothing was able to protect this binding site except for NAADP and ADP or unlabeled probe itself. Now, honing in a little bit more onto the selectivity, we conducted more photo labeling experiments, this time in the presence of increasing concentrations of unlabeled NAADP or unlabeled NADP. And here, as we quantify the level of protection of this binding site, we find about an 11-fold selectivity for NAADP over NADP. Now, we think this 11-fold selectivity is important because this is exactly the same level of selectivity that we had seen about 10 years ago with much simpler probes and previously published in our work in JDC and in Messenger. So now, at this point, we're feeling pretty confident that our new bifunctional probe is a high affinity selective probe for NADP binding proteins, and that human erythrocytes are a convenient source for working with these binding proteins. So now here's our probe in action. So we're starting with a human erythrocyte S20 supernatant fraction. We had taken this this protein through a series of chromatographic enrichment experiments, ultimately resulting in about a 200-fold enrichment of binding proteins. The protein was then photo-labeled with our probe. And on the left here is a silver stain, and on the right, a phosphor image. And you can see in this protein sample, to begin with, we have only a few protein bands present. And at this stage, our elusive binding protein is already covalently bound to our bifunctional probe. We then used click chemistry to biotinylate the probe, the ultraviolet agarose beads to capture and isolate that isolate that protein. We then very stringently washed these bead complexes and eluted, and then we go from these few bands shown here down to a single clean band at 23 kilodaltons. And this was notably biotin dependent because negative control reactions that were lacking this biotin azide came out completely clean. So we cut these gel bands out and we had submitted them for mass spectrometry analysis. And here is a summary of those mass spec results shown here. Now, this is a rank order of the relative abundance of peptides found in each of these proteins found in these biotinylated samples versus these original input samples. And you can see that there's one protein in particular that stands out that has about a 50-fold enrichment in our biotinylated samples compared to our input samples. And that's JPT2, which had exactly 0 peptides in our negative control samples. And in fact, JPT2 was so highly enriched in our biotinylated samples that we had over 90% sequence coverage of this protein in our mass spec analysis. So right away, we had a very clear front runner for a candidate binding protein. But what is JPT2? JPT2 is Jupiter microtubule associated homolog 2, and it's also known in the literature as hematological and neurological expressed 1-like. So this protein is actually very poorly characterized in the literature, but it does have a demonstrated role in cell growth and cancer. It's a short, hydrophilic, highly basic protein, and it has a couple of repeated sequence motifs found throughout the protein. And it's part of a gene family. So in humans, we have JPT1 and JPT2. Here's a bit of bioinformatic analysis that's done by Sandip Patel. Ultimately, in brief, we find that this JPT family is conserved evolutionarily, and it's present across a number of NADP responsive species. So now that I've revealed our candidate binding protein, we'll do some functional analysis, and I'll show you a series of experiments. So the first of which will look at ligand binding. So in order to directly ask the question, is JPT2 an NADP binding protein generated recombinant purified JPT protein? So here is a Kumasi stain of that purified protein. You can see a single band at 23 kilodaltons, nothing else present in the sample. And we incubated that protein with P32 NADP in the presence of increasing concentrations of competing unlabeled NAADP or NADP. So these reactions were then filtered through a 96-well PVDF filter plate. The plate was washed a few times, and then we incubated the plate on a phosphor screen. So this is a phosphor image of those actual binding reactions. So the P32 signal here is indicative of the amount of radioligand that's bound to JPT2. And you can see just visually that NAADP is able to protect this binding site with a greater affinity than NADP. And quantified here on the right, so we have about a 16-fold greater selectivity for NAADP over NADP with this protein. So these results suggest that JPT2 is indeed a bona fide NAADP binding protein. But does JPT2 regulate NAADP signaling? Well, I'll show you that firstly, it interacts with 2-pore channels. And secondly, its expression is an essential component of NAADP evoked calcium release. So in order to test whether it interacted with an ion channel, we conducted Co-Amino precipitation experiments. So here, these are HEC293 cells. We've transfected them with plasmids overexpressing GFP-tagged TPC1 or TPC2. We collected those cell lysates which had endogenous expression of JPT2. And here's an input Western blot just showing that we have expression of our GFP-tagged TPC fusion proteins. There's a GAP-DH loading control. And down below, this is endogenous JPT2 detected with an anti-JPT2 antibody. Now, when we immunoprecipitate JPT2 and probe for our GFP-tagged fusion proteins, we're able to detect TPC1 in our JPT2 immunoprecipitates. And reciprocally, when we immunoprecipitate our GFP-tagged TPC proteins using an anti-GFP antibody, we're able to detect JPT2 in our TPC1 immunoprecipitates. So these data suggest that JPT2 is indeed able to interact with an NADP-sensitive ion channel, and it seems to be biased towards interaction with TPC1. So next, getting to some functional data, with the help of Eugene Braileau from Temple University, we conducted microinjection experiments. So these are U2OS cells. They've been loaded with VERA2 calcium indicator. So Eugene injected an intracellular buffer as a control, and there's just a small injection artifact shown here. However, injection of NAADP elicited a rapid, robust signal. We can see here in blue. Now, if we transfected these U2OS cells with two different control non-targeting siRNAs, there's no effect at all on our NADP responses. But then knockdown of JPT2 with, again, two discrete siRNAs targeted against JPT2, we have a dramatic decrease in our NADP sensitivity. There's just a Western blot validating that these JPT2 expression at the protein level. Now, when we quantify these NADP signaling events, our JPT2 knockdown does significantly decrease NADP responsiveness, but also interestingly, injection of intracellular buffer into these wild type cells was not significantly different from injection of NADP into our JPT2 knockdown cells. So I think that really highlights that JPT2 expression is an essential component of NADP evoked calcium release. So next, we wanted to use a orthogonal approach to test whether or not JPT2 regulated NADP evoked calcium release, and so we wanted to look at a cellular process that NADP regulates, namely endolysosomal trafficking. So we employed a pseudovirus system, and this was kind of an extension of my thesis work published a few years ago, where in Cell Calcium in 2018, we had shown that coronaviruses exploit the host cell endolysosomal trafficking mechanisms during their infectious life cycle, and that inhibition of NADP TPC signaling could be used to prevent coronaviruses from translocating through this endocytic pathway. In order to run those experiments, I used a pseudovirus system, which essentially is composed of an HIV-1 backbone, and that's replication deficient, but it does have a luciferase gene insertion, which means we can assay for luminescence as a readout for viral infectivity. And these viral particles are pseudotyped with coronavirus spike proteins. These spike proteins are necessary and sufficient to mediate coronavirus cellular entry. So a few years ago, we were working with Middle East respiratory syndrome coronaviruses, but in the work I'll show you today, we're working with the SARS-CoV-2 pseudovirus. And SARS-CoV-2 is actually able to enter host cells in two different routes. So the first route would be through ACE2-mediated endocytosis, followed by endolysosomal transport of the virus through the endocytic pathway. And this, importantly, is the process that is regulated by NADP TPC signaling. Upon trafficking through the through the endolysosomal system, the virus eventually reaches a compartment where it is processed by host cell proteases, which prime and cleave the spike protein on the pseudovirus, triggering fusion between the viral membrane and the endocytic membrane, ultimately allowing translocation of the viral genome and luciferase expression. Alternatively, the virus can enter directly through the plasma membrane. So this occurs in tissues where they have high expression of TMPRSS2, the cell surface serine protease. So this protease can cleave viruses in the extracellular environment, directly triggering membrane fusion between the viral membrane and the plasma membrane. So you have direct translocation of the viral genome to the cytoplasm, and importantly, this completely circumvents the endolysosomal trafficking mechanisms and completely circumvents NADP-TPC signaling. So here in this experiment, we have HEC293 T cells. We've transfected with various siRNAs. We then infected the cells with our SARS-CoV-2 pseudoviruses. And here, as we go through that first route of endolysosomal entry in cells that are just naive HEC cells, we have a very low level of viral entry, but overexpression of that ACE2 receptor gives us a very high level of viral entry. Transfection of 2 non-targeting control siRNAs was completely without effect, but knockdown of TPC1 or TPC2 significantly blocked viral entry. As a control, we knocked down the lysosomal cation channel TRPML1, which also had no effect on viral entry, and neither did knockdown of JPT1, but knockdown of JPT2 significantly decreased viral infectivity. In contrast, if we overexpress the cell surface protease TMPRSS2 and we bias viral entry towards direct entry through the plasma membrane, then knocking down any of these proteins was completely without effect. So altogether, this data suggests that JPT2 is a regulator of endolysosomal trafficking. So here we've implicated JPT2 at the cell biology level into a pathway that's canonically associated with NADP-TPC signaling. So altogether, from these data, we can make these conclusions. We used a bifunctional photoprobe, AOIC NAADP, and that allowed us to purify JPT2 as a candidate NAADP binding protein. We then functionally validated JPT2, and that was done in a series of experiments, first through radioligand binding. Next, we showed that it's involved in NADP-evoked calcium release. And finally, we show that it's involved in endolysosomal trafficking. So with that, I'd like to thank our collaborative team. So our team include Eugene Brelu and Ellen Unterwald, who conducted our microinjection experiments, Sijun He and James Slama, who invented the AOIC photoprobe, which enabled us to do any of these experiments at all. Sandip Patel, who did all the bioinformatic analyses for us, and also thanks for organizing this event. Tim Walseth, who taught me how to do these photo labeling experiments and binding experiments, and has been the driving force behind the search for NADP receptors. And Jonathan Marchant, who's my mentor and advisor and has been endlessly supportive over the last decade or so. We've also been funded by these grants shown here. And if you're interested in the work that we've been showing here, our paper, we have a manuscript that's in press at Science Signaling right now. And on that note, we'd also like to thank Bjorn Philippe Dirks and Andreas Goose for sharing data over this last year. We've both been working towards the same protein here. And little shameless plug, in a few days, I'll be presenting the same data again at the Biophysical Society. So you can come see me again on Tuesday, the 23rd at noon. And with that, I'll take any questions.
Great, thank you. Great talk. We already have a couple of questions on the chat. So the first one is from Dejan Ren. Do you want to ask your question?
Yeah, it's very nice. It's fascinating that when you knock down JPT2 and a NADP-induced calcium rise seems to be gone. But we also hear from the previous talk, the same thing happens when you knock down the LSM12. Can you speculate what if could be any relationship between those two?
Sure. So, yeah, it's interesting. I guess from previous literature, we know that knockdown of TPC1 almost completely ablates NADP signaling, and we also know that knockout of TPC2 almost completely ablates NADP signaling. And so here we're showing kind of a biased interaction of JPT2 with TPC1, and I know that there's a lot of data that the previous group has shown of LSM12 interacting with TPC2, so it could be that knockdown of either kind of redundantly inhibits NADP signaling. We do have some somewhat preliminary data that might be of interest. Let's see. So we had come across, let's see, 1 second. So we'd come across the LSM12 preprint, which we thought would be very fascinating on bioRxiv. And so we wanted to test LSM12 in our own hands. So here we have, this is a Co-Amino precipitation experiment looking at both LSM12 and JPT2. So we have, these are U2OS cells overexpressing TPC1 or TPC2 GFP. This is endogenous LSM12, endogenous JPT2. And when we immunoprecipitate our GFP tagged TBCs and blot for either of them, we do find kind of a bias for JPT2 to interact with TPC1, but there does seem to be a little bit of interactivity with TPC2. And LSM12, we're now confirming the previous group's findings where we see an interaction with TPC2, but and a little bit less of an interaction with TPC1. So it could be that they're perhaps both cooperatively involved in NADP signaling.
All right, thank you.
Yep.
Great. We have a second question from Gert Boltinik, if you want to ask your question.
Yes, actually, I have a different question than the one by listening to this. If you knock out GPT-2, do you lose LSM12 in these experiments or oppositely?
That's a good question. Yeah, so we haven't taken a look at this sort of, I guess, are you thinking of compensation if you knock out, if you knock down JPT2, do you see an increase in LSM12 expression and vice versa? So that's could be a pretty provocative idea. It's not something that we've looked at quite yet, but I'll add it to the list of experiments to do.
Yeah, it's all very fresh, but when you presented these data here, this was what popped up. I mean, my originally phrased question was about whether GPT-2, about its expression among cell types and tissues, whether it's ubiquitously expressed or there is some, distinct patterns.
So,
maybe you mentioned that and it escaped my mind, but.
So here is a analysis pulled from the Human Protein Atlas. So this is, excuse me. Sorry. So this is an analysis of cell types just on the Human Protein Atlas. And so you can see that it's expressed across a very large number of cell lines. And we're highlighting here 2 cell lines, SKBR3 and U2OS cells, that are well characterized in the literature as being NADP responsive cell lines, and we have very high expression of JPT2 in these cell lines. As far as tissue distribution, we know that it is pretty much ubiquitously expressed. It does have higher expression in some tissues than others, I think, namely liver, pancreas, spinal cord. I guess I'll note that the original TPC papers with TPC2 knockout in pancreatic beta cells, that's an example of NADP sensitivity in pancreas, and here we have very high expression of JPT2 in pancreas. Similarly, there's been work on non-alcoholic fatty liver disease in liver, and of course JPT2 is highly expressed in the liver. I think JPT2 is also highly enriched in cancer tissues compared to normal healthy tissues.
Okay, thank you very much. Very fascinating. Great talk, by the way.
Thanks.
And we'll go with one more question, and then we'll have to move on with the next talk. So Anthony Morgan or Jan, not sure who's asking questions.
This is Leanne with a very deep voice.
Yeah. Okay.
So fantastic piece of work. Really great. Now, you started off saying that the Jupiter protein was cytosolic. So can you observe any dynamic interaction or coming to the endolysosomes when NAADP levels go up, for example?
That's not an experiment that we've done yet, but it's something that we really want to test. So I guess the experiment would be adding perhaps NAADP AM and having a fluorescently tagged or looking fluorescently tagged GPT-2 and seeing if it then aggregates in membrane organelles like endosomes or lysosomes. So I think that's on the list of experiments to do. At this point, we're just excited with this new discovery, and now we've opened so many doors of future directions.
Okay,
so,
thank you.
Don't have an answer for you yet.
Yeah, no, that's great. Thanks, great.
Great. Thank you again for your great talk.